Hi dear students, welcome back to one more session of immunology. In the previous class we had mentioned about the adaptive system and we came across that mainly cellular factors, especially the T lymphocytes and the B lymphocytes are the ones which are being responsible for the adaptive or the acquired immune system. Today we would be talking about what are the characteristics of these lymphocytes, what are their surface markers and what are their functions. The two main lymphocytes which take part in the acquired immune system is the B lymphocyte and the T lymphocyte. The B lymphocyte, as we all know, they mainly produces antibodies within our body in response to the antigen exposure, as well as T lymphocytes are mainly involved in the cell-mediated immunity. That is, they do not provide any antibodies, but instead they carry out various other functions in which they directly interact with the antigens. So let's go into the details of that. How and when these two lymphocytes act. If you go to look onto the surface of a B lymphocyte, you can see attached to the B lymphocyte there is a B cell receptor. And on the surface of the T lymphocyte, you have a T cell receptor. It is based upon these receptors that we help to distinguish between the function as well as the role of the lymphocytes. The B cell receptor somewhat looks like the antibody which has been produced by the lymphocyte. So let's go into the details of that. If you go to consider the B lymphocyte is mainly being produced and matured in the bone marrow. Bone marrow is a primary lymphoid organ and the synthesis as well as the maturation of the B lymphocytes mainly happen within the bone marrow. And as I mentioned earlier, the B lymphocyte is mainly involved in the humoral immunity or the antibody mediated immune response. And it does have lots of surface molecules attached onto it. And one among this, there are lots of receptors and you can see one of these receptors over here is IgM. This is a immunoglobulin or an antibody. And another ones which we come across is the class 2 MHC. Okay, what is that? I will be just mentioning you. So you have class 2 MHC, you have CD40, CD21, CD45, like that. There are different receptors attached onto the B lymphocyte. And it is all these receptors which will help the B lymphocyte to interact with the antigen and at the same time carry out various other functions which is promoted by the B lymphocyte. The B lymphocyte, it is mainly produced as well as matured in the bone marrow. This we had mentioned earlier. So when it becomes mature, it does contain all the receptors which have been needed for its function. Like it will have antibodies attached to it. It will have different class 2 MHC molecules as well as different receptors constituting the B cell receptors complex which that would be present on the B lymphocyte. And till interacting with an antigen, the B cell does not becomes activated. Only when an antigen or especially when the soluble antigens come and interact with the B cell, only then it gets activated. Till then, it is not producing any antibody into the blood. So when an antigen comes and interacts with the antibodies, I had told you about here, the antibodies or the IgM as well as IgD, these are immunoglobulins or antibodies which have been attached onto the B lymphocyte. So when such antigens come and interact with the immunoglobulins attached onto the B cells, what happens? The B cell gets activated. And at the same time, some of this antigen is also being processed by the B lymphocytes and it is being presented by the B lymphocyte through another molecule called the MHC molecule. Okay, and this MHC MHC bearing antigen is being recognized by the T lymphocyte, a type of T lymphocyte called the T helper cells. So during the activation of the B lymphocyte, what happens? You can see that the B lymphocyte is also giving a small portion of the antigen attached to its one receptor called the MHC, which is the MHC here. You can have a look. The class 2 MHC attached onto the B lymphocyte, and it is providing that to the T helper cell also. Now, once it gets activated, what happens? The B lymphocyte will produce some cytokines and all that. Cytokine receptors will be produced. Cytokines are signaling molecules, chemical molecules which have been produced during the activation of cells. And what happens? The B cell, the activated B cell, will get differentiated into two different type of cells called the plasma cell and the B memory cell. Now, what is the role of this plasma cell? The plasma cell is the one which produces the antibodies. So, once an antigen happens to bind with a B cell type of B cell receptor, what happens? That B cell becomes committed to produce antibodies of that particular type. So, now if dengue antigen is coming and binding onto this and the B cell gets activated, what happens? This cell becomes committed to produce antibodies against the dengue virus alone. So it will be the plasma cell which will be producing the antibodies, and a memory cell, a memory B cell is also being formed which bears the antibodies which are being specific to the dengue virus. So each time a different type of antigen comes, different types of plasma cells and different types of memory cells are also being created specifically against it. Now, if the activation signal is not proper, or if it has been found to be inappropriate, or proper activation is not happening, what happens? The B lymphocytes, which was previously tried to be activated, will undergo cell death by a process called apoptosis, or the programmed cell death of that does happen. So this is the process of B cell activation which happens. So the B cell interacts with the antigen and it also processes the antigen and presents it to the T helper cell, or which is a type of T lymphocyte, and it gets activated. Once it gets activated, it forms the plasma cell and the memory cell. Now, it is the plasma cell which secretes immunoglobulin, and it is very rarely seen in the blood, but it is mainly found in the lymph node as well as in the spleen. And the memory B cell retains the memory of this interaction with that particular antigen. So now if we go on to look onto the B cell surface molecules, you can see that we do have the membrane bound immunoglobulin that is a part of the B cell. Then in addition to that, we can see that the antigen recognized by the B cells immunoglobulin is presented in class 2 MHC to the T cells during the B cell activation. This is the receptor which we show here, the class 2 MHC here. The class 2, through the class 2 MHC, it will show it to the the T helper cells. Then you also have CD35 and CD21 receptors where the complement factors can bind once the complement is activated, because the immune system is totally linked. It is not one cell alone which helps us to fight against an antigen. Different molecules interact. So on the surface of the B cell itself, there are different markers. And CD80 and CD40, they will bind to the T cell proteins and they provide the co-stimulatory signal in the B cell activation. Then CD32, that's another receptor for the immunoglobulin binding. Okay, immunoglobulin binding, this is CD32 here. You need not worry about all these receptors, but just make sure that there are lots of receptors attached to it. Then CD45, it is a protein tyrosine phosphatase receptor. It regulates the activation of B cell and T cell activation. And then we have CD54, which is an addition molecule involved in the helping the B cells to bind to other cell types. So the main receptors, do remember, you have the immunoglobulin receptors, IgM as well as the IgD, and you also have a class 2 MHC. What is MHC? It is called the major histocompatibility complex. I will be explaining what is that in the coming sessions. So it is a one which helps to interact with the T helper cell, to the T cells. Now, if you go on to the functions of the B lymphocytes, you can see that the antibody production is the main function of the B lymphocyte, and at the same time, it will act as an antigen presenting cell and present the antigen to the T lymphocytes that we had seen in the previous thing. That is, you can see here, in this process, it is presenting the antigen to the T helper cell. So, the second function of the B lymphocyte is to act as an antigen presenting cell and to present the antigen to the T lymphocytes. And at the same time, it provides the signal for the T cell activation. So these are the three main functions of the B lymphocytes. Now let's move on to the next one called the T lymphocytes. The T cells are the cells of the cell-mediated immunity. Now, the T lymphocytes, they are also being formed from the bone marrow, but they mature within the thymus, and that's why we call it as a T lymphocyte. And like the B lymphocytes, these cells also do have membrane receptors for the antigen. But although the antigen binding T cell receptor is structurally distinct from immunoglobulin, it does share some structural features with the immunoglobulin molecule, most notably in the structure of the antigen binding site. And you can see that unlike the membrane bound antibody B cells, though the T cell receptor does not recognize a free antigen, instead the T cell recognizes only the antigens that is bound to a particular type of self molecules. And most T cells will recognize the antigen only when it is bound to a self molecule encoded by genes within the MHC. So let me just make it more simple for you. If you go to consider the B lymphocytes, all the soluble antigens which is being present of the pathogen present in the blood can directly come and interact with the B cell receptor or the B lymphocytes. But in the case of T lymphocytes, soluble antigens of the pathogen present in the body cannot directly come and bind onto the T cell receptor. Instead, these molecules can be only recognized if certain self molecules, that is, if the host, if I am a host, some molecules which are being produced by my own genes should present these foreign pathogen to the T cells. Only then the T cells can identify it. And those self molecules of my or your host gene which will present the foreign antigen to your T cell is referred to as a major histocompatibility complex. In simpler words, major histocompatibility complex is a set of genes which will introduce or help us to introduce the foreign antigen to the T lymphocytes. It's like a mediator. Only if the MHC complex will process and present this gene, present the foreign antigen on its surface, only then the T cells can recognize this. So, to be recognized by most T cells, this antigen must be displayed together with the MHC molecule or the major histocompatibility molecules on the surface of the antigen presenting cells or on virus infected cells, cancer cells, grafts and all that. So that is a reason why you had seen earlier when it was being presented to the T helper cell, the class 2 MHC which was being present on the B cell was presenting the antigen to the T helper cell. So do remember the T cells or the T lymphocytes, they cannot directly interact with the antigens, but they can only interact with the antigens which have been displaced by the major histocompatibility complex. And based upon this, these functions will contribute towards the uh, T cell receptor complexes. As I told you, it is formed in the bone marrow and it matures in the thymus. And the T cell receptor is found to be specific, and it will recognize the MHC bound antigens. Now, all the T cells expresses an antigen binding T cell receptor and CD2 and CD3 glycoprotein on their cell membrane. There are lots of proteins or receptors attached onto the thing, and most of the T cells, they are distinguished based upon the presence of CD4 and CD8 glycoprotein uh, receptors on their cell membrane. Now you might think, what is this CD4, CD8 and all that? Now if you go to consider uh, different types of proteins which have been present on cells. Like that, on the surface of T cells, there are certain molecules, okay, glycoprotein molecules or glycoprotein receptors, and based upon their type, we are naming them as different. So most of the T cells, they have CD4 and CD8 glycoprotein receptors based upon their cell membrane, and based upon if they have a CD4 receptor, you call it as one particular type. If you have a CD8 receptor, you call it as another type. So the CD4 bearing T cell receptors, they are referred to as the, you can have a simpler look over here. If you have a CD4 receptor, you call it as a helper T cell. And if you have a CD8 receptor, you call it as a cytotoxic T cell. And sometimes you will be having a CD4 receptor itself and a CD25 receptor. Then you call it as a regulatory cell. Okay, but in all the T cells, the T cell receptors are common. That is, these are the ones which will interact with the MHC molecules. Okay, so based upon the surface molecules, the T lymphocytes itself is being divided into different types. You can have T helper cells, T cytotoxic cells, as well as the T regulatory cells. The T helper cells, they have the CD4 receptor on them, and they are activated by the recognition of antigen on the class 2 MHC on the antigen presenting cell. You do remember on the B lymphocyte you have the class 2 MHC molecule, and so when the antigen is being presented on the class 2 MHC of the antigen presenting cell is interacting with a T helper cell, it gets activated. And once this gets activated, it becomes into the effector T helper cell, and it will secrete various cytokines which will help the B cell activation for the production of antibodies. And on the cytotoxic T cell, they have a CD8 receptor, and these are the ones which will interact with the class 1 MHC. Class 1 MHC, that is, certain particles are there present in on all cells we have the class 1 MHC, and these are the one which will help us to recognize whether it is self or not. So when a cell is getting altered by the either by tumor or virus infection, the class 1 MHC will get changed, and so what happens is that class 1 MHC is been interacting with the T cytotoxic cell, and the T cytotoxic cell will understand that the class 1 MHC is being found to be changed, and it tries to kill that particular cells which are being changed. So when an antigen binding on the T cell receptor of the CD8 cell, it is activated and it divides into clones of memory and the effector cells, and the effector cytotoxic cells is known as the cytotoxic T lymphocytes, which kills the viruses infected onto the cell and the tumor cells. Then we have another type of molecule called the T regulatory cells. or the T reg is also called as a suppressor cell, and it helps to suppress the humoral and the cell mediated immunity. So to control the activity of the what is it of the T lymphocytes, we have the T regulatory cells. uh, you need not get confused because of there are lots of receptors on the T cells, but do remember the most important among them are those receptors which will interact with the MHC complexes. What are MHCs? MHCs are certain molecules which have been produced by our self genes, which will help us to recognize between self and non-self. So there are basically two types, class 1 MHC and class 2 MHC. Class 1 MHC is mainly being presented on all nucleated cells, and class 2 MHC is mainly being found on the antigen presenting cells. So the T helper cells, they do have a marker called the CD4 receptor on their surface, whereas the cytotoxic T cell, which kills the cells, it mainly produces a CD8 receptor. And commonly speaking, what happens? The T helper cell, the cytotoxic cells, and the T regulatory cells do have the common T cell receptors. Now, the T cell receptor of the helper cell, it interacts. The CD4 bearing T helper cell interacts with the class 2 MHC, and the CD8 bearing cytotoxic T cell interacts with the class 1 MHC. You can just remember this formula: 4 into 2 is equal to 8 into 1. That is all same. So the CD4 bearing T helper cell interacts with class 2 bearing MHC, and the CD8 bearing cytotoxic cell interacts with a class 1 MHC. So you got an idea of the different types of T lymphocytes. So we have T helper cells itself. There are subclasses T helper 1 and T helper 2. They have some T helper 1, it mainly produces cytokines which are involved in the inflammation, as well as for the activation of T cells and macrophages. And the T helper 2, it mainly participates in the activation of B cells and the immune response. And TC cells, or the cytotoxic T cells, they are the ones which interact with the class 1 MHCs present on the surface of an altered cell, and they mainly produce cytotoxic substances, and it gets differentiated into the cytotoxic T lymphocyte once it interacts with an altered class 1 MHC. So they will kill the altered self cell. Then we have the T suppressor cell, which will regulate the immune system. So the functions of the T lymphocytes, it will help in the B cell maturation, expression and antibody production, as we had seen the T helper cell is doing it. They help in the recruitment and activation of mononuclear phagocytic cells. Mononuclear phagocytic cells, they mainly include the antigen presenting cells. So for the recruitment of and activation of these mononuclear phagocytic cells, the T cells do play an important role. They help in the recruitment and activation of specialized T cytotoxic cells in antiviral response. They secrete cytokines which are responsible for the growth and differentiation of the T cells, monocytes and macrophages, and also they help in the regulation of the immune reactions. I would also like to talk about the null cells. These are granular lymphocytes. These are also a type of WBC, which will display cytotoxic activity with a wide range of tumor cells in the absence of any previous immunization with the tumor. And it is the null cells. Why they are called so? Because they do not have a T cell receptor, or neither do they have any immunoglobulin incorporated in their plasma membranes, but they recognize the potential target cells in two different ways, and they form different type of cells called the NK cells as well as the killer cells. We have already mentioned about the NK cells in the in the inborn or the innate immunity which we had. I just mentioned it here because this also do take place some role in the acquired or the adaptive immunity also. And the null cells or the NK cells, they employ receptors to distinguish the abnormalities especially a reduction in the class 1 MHC molecules and the unusual profile of surface antigens displayed by some tumor cells as well as cells which have been affected by the viruses are being detected by the null cells. And another way in which the NK cell recognizes the potential target cells depends upon the fact that some tumor cells and cells infected by certain viruses display altered antigens, and some of them may the immune system has made an antibody response, so that the tumor or the antiviral antibodies are bound onto their surfaces. And because NK cells express a CD16, a membrane receptor for the carboxyl terminal end of the IgG molecule called the Fc region, they can attach to these antibodies and destroy the target cells. So they also carry out a process called the antibody-dependent cell-mediated cytotoxicity. I would be going into the details of this in the coming sessions. This is just just an introduction of it. So thank you for now.